Hello and good evening everyone. Welcome to March's edition of the Atom Society live streaming events. I hope you are all doing well and you've been getting out in the sunshine and seeing the crocuses and the daffodils and all the lovely spring flowers coming out. We've got a really interesting talk lined up for you this evening, and we will get onto it very soon. A few housekeeping things beforehand. I mentioned last month about the Atom Festival. So that is going to be starting on the 11th of June in whatever form it does happen. I've got my notes in front of me this time, so I'm not getting confused between the dates. The festival committee are not sure yet what form it will take, and they can't really make any decisions on that at the moment. However, in the event that there are in-person events, it may be that the committee needs some volunteers. And so Susan Armstrong has asked that anyone who would be willing to volunteer, please contact, you can contact me with the Atom Society Secretary email account, or if you know Susan, contact her directly. And so we can let her know that you would be available for volunteering and what you would be able to do. As I said, it might not be needed because it might be that everything needs to be done online, but just in case, they are calling out for volunteers to be prepared. Nothing I wanted to bring up with you all. Obviously, the committee works hard throughout the year to try and make sure that you have an interesting and varied program of talks. We often get suggestions from members about speakers to invite, but we were wondering if there was any particular areas or themes or topics that you would like to hear an expert come and speak about. The committee have got their own ideas, and they'll be collating them at the next committee meeting and discussing what kind of areas that we would be interested in hearing from someone, but we wanted to know from you, is there something that you would particularly like to hear an update on, for example? So yeah, let me know. You can email myself or anyone on the committee that you're in contact with and let us know, and we'll see how we can try and fit that into our program. So before we move on to the main event, we have our ever popular science snippets. I know that the committee have got a few lined up, so we will go to those first. But if any of you guys have any, as always, please pop them in the comment section on the YouTube channel, and we will bring them up here. So I will bring James in first, who said he had a couple. Hi, James.
Hello. Good evening, everybody. Good evening. Yes, I've got a couple of snippets. Some of my friends know that I'm quite interested in all matters space, and somebody phoned me today and said I should listen to a particular piece on a NASA website, which I did, and it was the sound of this wonderful Perseverance rover crunching over the gravel on the surface of Mars, and you could hear this thing going over the gravel, and that's amazing to be able to hear this sound from Mars. But the sound was a bit odd. I was thinking to myself that if your car made a sound like that, you'd have it into the garage for a thorough overhaul as quick as you possibly could, because it would sound dangerous. But it was fascinating to hear the sound from Mars. The second snippet I have is one to do with some correspondence that's been going on for some considerable time in some of the journals, to do with SETI. I'm sure you know what SETI is, the search for extraterrestrial intelligence. You haven't found much yet, as far as I know. But one of the ways of searching for it is to see whether there's any signs of other solar systems showing indications of superintelligent activity. And one thing is what's called a Dyson Sphere. You may have heard of a Dyson Sphere. And the theory is that some intelligent creatures on some distant solar system have been able to tap into the energy of their local sun by building a metal sphere right around the sun, a hollow sphere around the sun, which captures all the energy, and they can then use it on their little planet for central heating or charging up their mobile phones or whatever it may be. Now, the correspondence was about saying, well, no solar system would have enough material in its planets to build such a thing. That isn't true. If you take mercury, for example, and I'm a maths graduate, but this is only A-level maths. Take mercury, imagine you flatten it to a thin sheet or to a sheet and form it into a sphere, put it around our sun at maybe 100 million miles from the sun. We're 90 million miles, so it's outside our orbit. How thick would the shell of that be? Well, I've worked it out, and I thought you'd like to know. It's 86.23 centimeters.
Okay.
Yeah, so you could do the whole thing with mercury. Anyway, those are my two snippets, so I'll now pass you back to Danielle. Okay, thanks.
Thank you very much, James. Okay, now I'm going to bring Alphonse in at the end because I think he might have something exciting for us related to what James was just talking about, but I'll bring Peter in now.
Hi, everybody. Hope you can hear me.
Yeah.
Yep, good. So yeah, I was interested in a little thing that I found in The Economist describing a UNESCO report, which was published right at the beginning of this month about blue carbon, which is carbon storage by things underneath the sea surface, at least some of the time. So these are meadows of seagrass, mangrove swamps, and tidal marshes, which act as sinks for carbon dioxide. And across the surface of the world, it's estimated that they capture or are responsible as a sink for as much as 10% of the carbon dioxide that needs to be captured from the gases and global warming over compared to the level that there was in the year 2018. So these are really important ecosystems that need our protection and expansion as much as they can. That's what I had, and Danielle is showing the link for the report and the front cover of the report. Yeah.
Yeah, I can pop the link in the YouTube chat.
Thanks.
As well,
Thanks.
Everyone can access it. Thank you very much, Peter. Okay. Alphonse, I think you have a few snippets.
Now if
oh, was that it? Sorry.
No, it goes on. At least it should go on.
It stops now. There we go.
Yeah, that was actually the sound that let me stop this. That James mentioned earlier. So if he wouldn't have mentioned it, that would have been one of my science snippets. The other one I have is actually this picture. Do you know what that is?
No.
It's one of the world's most sophisticated incubators. And what people did there is, for the first time, they managed to grow embryos, and these are mice embryos in this incubator. So not having to have a womb, in like this entirely artificial incubator, you were able to grow mice up to the 7th day of their pre-life, if you want.
Wow.
So that was quite a revolution. And okay, you don't grow humans in there, but at least having mammals at that stage is something quite new, which hadn't been achieved before.
Definitely. That's really cool. Thanks very much. Is that your last one, Alphonse?
Yep, that was the last one.
Fabulous. Okay, so it doesn't look like anyone from the membership is offering any up, but obviously if you do think of one, please feel free to pop it in the chat and we can bring it up later. So what I will do now is I will bring James back in and he can introduce this evening's speaker.
Yes, hello once again, everybody. Our speaker tonight, Zahra McVey, I met her when she came as a guest to one of the Atom talks in not that far back, 2019, when we were meeting at the King's Head and Bell. Some of you will remember those far off days. It seems a long time ago now, doesn't it? And hopefully we'll be back not there, but at another venue before too long. And Zahra was one of those people that came along. We got chatting, and she came across as a very interesting person and an interesting subject she was working on. And it's happened before when people come along like that, and we say, "This is fascinating. Would you be consider giving a talk to the society?" And happily for us, she was willing to do so, and she did. Just as an aside, she wasn't the only one, because another person I remember coming along as a guest and me having a chat with her when I was then chairman and saying, "This is very interesting. Would you be prepared to give a talk?" And she said yes. And she did. And not only that, she became a very active member of the society, became our secretary, and is now our chairperson. So it just shows where you can go if you come along to the society. Anyway, back to Zahra McVey. Her subject is computational biology. Now, I, by my early degree, was a mathematician, and I knew what maths was, more or less. But mathematics and biology, do they mix? Chalk and cheese? So I was intrigued to find out what computational biology was all about. And Zahra is the person to tell us. And it sounds, from what I've heard so far from her, and I've had a chat with her, absolutely fascinating. She works with large data sets, which didn't exist some years ago, in my early days, involved in a lot of modeling and so on, and is looking at various diseases, and trying to find ways of developing drugs which will attack those diseases by modeling and simulations and so on. It sounds incredibly clever. Well, I'm not going to attempt to describe anything which she does. She works for Novo Nordisk, been there for 2.5 years or so. It's in the depths of the research in the subject, world-leading stuff. She's worked in Copenhagen for the company and now works happily for us in Oxford. So without further ado, I am delighted to introduce to you to Dr. Zahra McVey. Zahra, over to you.
Hi, thank you for the kind introduction and the opportunity to share some of our work with you all. As mentioned, I work as a research scientist for the pharmaceutical company Novo Nordisk, and today I would like to discuss how we can use this topic of computational biology in drug discovery. We'll start off with a brief introduction to myself and R&D at Novo Nordisk. We'll then move on to defining what multi-omics is and how we can use it in discovery. I'd then like to touch on how we're approaching the discovery of novel small peptides for target discovery. And finally, I'd like to share some of my thoughts regarding drug target prioritization with you. Starting off with a little bit of background about myself, I started off as a biochemist, completing my undergraduate degree at Royal College London. I stayed on there to complete my master's in molecular and cellular biosciences, which was my first real experience working in a wet lab doing research. I then moved to Oxford, where I completed my DPhil at the Weatherall Institute of Molecular Medicine, for leukemia stem cell biology. It was during my DPhil where I discovered my interest both in bioinformatics and data analysis, but also clinical application of my work. That's why I then decided to move to Copenhagen, where I took a position as a big data analyst, working for the company Novo Nordisk. And most recently, I transitioned to a role of a research scientist in a research site, still for Novo Nordisk, but based in Oxford. Novo Nordisk is a global pharmaceutical company headquartered in Denmark with affiliates in over 80 countries. It is among the world's 10 largest pharmaceutical companies, having founded in 1923. Novo Nordisk has a strong history of working in diabetes, but also is developing other serious chronic diseases such as obesity, and also in rare blood and endocrine disorders such as hemophilia. Novo Nordisk R&D is spread globally across many R&D sites, located in Denmark, the US, now in the UK, and also in China and India. R&D has the main ambition of innovating to improve patient outcomes, and they do this in 6 main therapy areas, which are diabetes, obesity, rare blood and endocrine disorders, cardiovascular disease, and non-alcoholic steatohepatitis, a disease of the liver. If we take a more close look at Novo Nordisk's R&D strategy, this is based on three main strategic focus areas. Firstly, this is expanded into other serious chronic disease areas, and this includes the mentioned, but also things such as chronic kidney disease and brain disorders. Secondly, this is thinking about externalization, and this is an ambition to increase our external collaborations, both with academic researchers, but also with biotech companies. And finally, there is an aim to raise the innovation level we have in R&D, and that's everything from exploring new biologies, but also moving into the use of new technologies. Historically, Novo has a very strong world-leading profile in the development of therapeutic proteins, typically in injectable uses, but we're now also exploring the use of and oral therapies. We have a strategy for us to be able to take a new medicine from the idea to a patient, but we know this is a long and a costly process. An idea needs to start in the exploration phase in early research and then needs to go through some preclinical testing. If successful, it will then go through an upscaling process where it is then taken into clinical trial. Again, if successful at clinical trial, it will then be taken to product registration with the appropriate authorities. This is, of course, done by our colleagues in the commercial area. We need to pave the way in the market for our new drugs. Now, historically, out of roughly 10,000 ideas, only one will potentially reach the market. And this has typically taken 10 to 15 years for this process. And this highlights the incredible growth we've seen recently in taking a COVID-19 vaccine to the market in record time. And of course, now we're building on new technologies and drug discovery methods to try to increase the speed of this process and be able to increase the success rate. If we now turn to Novo Nordisk research in Oxford, the new research is based on the Old Road campus in Oxford, and we have amazing neighbours ranging from the Big Data Institute to the Wellcome Centre for Human Genetics. We have a multidisciplinary approach ranging from the Computational Biology team, which I am located in, to human genetics, and then working with our colleagues from our Discovery Technologies and Genomics team, and also our Discovery Biology and Pharmacology. We have a strategic focus to collaborate both with academia and industry. And we work across the cardiovascular and metabolic disease space to try to go into blue sky high risk areas that previously haven't been explored at the company. I'm now going to move on to defining what multi-omics is and how we can try to use it in drug discovery. We're going to start off with some fundamental principles and build onto that some more complex methods. The central dogma of biology is that DNA makes RNA and RNA makes protein. Our DNA is the genetic material that we inherit from our parents, and it encodes all our basic characteristics such as our hair color and our eye color. In order to do this, our DNA is split up into functional units called genes. In order to utilize these genes, our DNA is converted into a molecule called RNA through a process called transcription. This RNA contains the instructions to generate our final product, protein, which is the functional unit of all our cells. The process of generating RNA to protein is known as translation. DNA and RNA is made up of a 4-letter code: A, T, or U and R, C, and G. And proteins are made up of building blocks called amino acids. The large-scale study of our DNA is called genomics. The large-scale study of our RNA is called transcriptomics, after the process of transcription. And large-scale study of our proteins are called proteomics. Collectively, these are known as multi-omics. But the multi-omics field is greatly increasing, And this is now in other fields as well, such as epigenomics, which includes DNA modification, microbiomics, such as our gut microbiome, and metabolomics, which consists of small metabolites, for example, found in our urine. I'd like to now look at how essential our genetic code is and how a mistake in this can lead to a disease. Hemophilia B is a serious blood clotting disorder. In healthy individuals, we have a gene called the F9 gene. This encodes for this messenger RNA, this transcript, called the F9 transcript, which carries the instructions to generate the protein called coagulation factor 9. This coagulation factor is essential for maintaining healthy blood clotting in our bodies. Unfortunately, in some individuals, there is a genetic mutation in this F9 gene. The full code is no longer correct. Therefore, the RNA instructions are not correct for generating our factor 9 protein. This results in either an absence of the production of factor 9 or an abnormal protein. This then leads to these patients experiencing abnormal or no blood clotting. We can see how there are multiple ways for treating these patients here. We're either to the problem, and we can treat the patients with replacement factor 9. However, this will need to be administered multiple times, or we can go to the root of the problem and try to fix the F9 gene through gene therapy. Hemophilia B is what we call disease, where one gene.
Sorry to interrupt you.
Yes?
The sound, we're having a bit of an issue with the sound. Are you connected by Wi-Fi? Is it possible to plug in? So I wonder, or because it feels like it's a connection issue.
I am on Wi-Fi. I'm not sure I can connect to it though.
Okay. It seemed to be okay before, but it's a bit laggy now.
Okay, I can try going onto a wireless hotspot. That may help things.
Okay, yeah, maybe we'll try that. If that doesn't work, we might just have to switch off your video for the talk. Maybe.
That's fine. I'll just switch to hotspot now.
Are you still there? You look like you're frozen. Oh, okay. Oh, you're back.
Hi, is that any better?
That's much better. Yeah.
Okay, we'll stick with that then.
Okay, I'll let you carry on. Sorry about that.
Okay, no problem. So we were just discussing how hemophilia B is a monogenic disease, and this is where we have one striking mutation that is able to cause one disease. However, in many complex human diseases, such as metabolic disease, we know this isn't the case. Here we know there is a genetic predisposition to the disease, but the picture is a little bit more complicated. In order to study this, we need to gather individuals with type 2 diabetes, for example, and healthy controls. We then isolate a sample of DNA from each group of individuals. We then look at the genetic mutations across all of these individuals' genomes, and this is a process known as genome-wide genotyping. This allows us to ask the question, do patients with type 2 diabetes have different genetic mutations when we compare them to their healthy controls? And the method we use to answer this question is called a genome-wide association study. The results of which are shown in the plot below. This is termed a Manhattan plot due to the resemblance of a Manhattan skyscraper skyline. On the x-axis, we have all of the genetic mutations ordered by chromosome, and on the y-axis, we show the significance of their association with type 2 diabetes. And as you can see, there are many dots or genetic mutations that potentially have a significant association with the disease. And I'm sure you can imagine that genes mutated in type 2 diabetes are potential candidates for further drug discovery. I'd like to now move away from DNA to thinking about how RNA can be used to switch genes off and on. If we take the example of two cell types found in our pancreas, pancreatic beta cells and pancreatic alpha cells, we know these cells have very different biological functions. However, both cells contain exactly the same DNA. If we take the example of the INS gene that encodes the hormone insulin and the GCG gene, which encodes the hormone glucagon, both of these DNA sets are found in both of these cell types. However, a beta cell only needs to produce the hormone insulin, and an alpha cell only needs to produce the hormone glucagon. Therefore, these cells switch on the production of RNA only from the gene which they require. So the insulin transcript, or mRNA, will only be produced in the beta cell, and the glucagon transcript will only be produced in the alpha cell. This then allows the insulin protein to specifically be produced in beta cells, and the glucagon protein to be specifically produced in alpha cells. Therefore, these two cell types are able to carry out their differential functions of beta cells using insulin to decrease blood sugar, and alpha cells using glucagon to increase blood sugar. So we can see how the RNA present in a cell is like a fingerprint representing that cellular function. We can now move on to how we can use that fingerprint of RNA to look at disease and dysregulated gene expression. So for example, now we can start with biopsies from healthy livers, and we can compare them with diseased livers with the disease NASH, as mentioned earlier. We can collect RNA samples from both of these tissue types, and then we can decode this RNA sequence in a process called RNA sequencing. This allows us to ask the question, do patients with NASH have different liver gene expression profiles when compared to healthy controls? The results of which are shown in this plot, and this is termed a volcano plot because of its resemblance to an exploding volcano. And on the x-axis here, we have the log2 fold change, which represents the change in a gene's expression in the disease state, so NASH compared to healthy. Therefore, genes with a positive fold change here are more switched on in NASH, and genes with a more negative fold change are more switched off in NASH. And the y-axis here again is indicating the significance of this association and therefore our confidence in it. And again, you can see how genes that are differentially switched on and off in a disease state are potential candidates for drug discovery. But we can actually take this one step further, because the liver tissue is composed of many cells and many cell types. If we go back to our example of the pancreas, we've already seen how a pancreas has both beta and alpha cells with very different functions. If we average out the gene expression across this entire tissue, we see an average signal which is summing together all of those differences we see across the cell types. So what we really need to do to disentangle this is to now look at the switching on and off of these genes at a single cell resolution. And this is something we're able to do. So in this example, we can take healthy pancreas, and we can take pancreases from type 2 diabetics. We can take these tissues and dissociate them to a single cell suspension, where we then isolate the RNA from each individual cell. We can then do sequencing and decoding of the RNA in each of these individual cells. And this allows us to now ask the question, do single cells from both healthy and type 2 diabetic pancreases have different gene expression profiles? This is a huge amount of data, but one way in which we can look at this is through dimensionality reduction plots. This is a 2D representation of all the variation across all cells, across all genes. Each dot on this plot represents one cell, and the dots will form clusters of cells based on similarities in the genes that they are switching on and off. So for example, here now we can see our beta cells coming together here on the left that are functionally distinct from the alpha cells, which are coming together here on the right. And if we look at the bottom, we can now look at our glucagon and our insulin genes, and you can see the glucagon gene, based on its red expression, is over here in the alpha cells, as we would expect. And the insulin gene, based on the red color, you can see has high expression in the beta cells, again, as we saw in the example earlier. But we can also compare the gene expression across different cell types and across the different conditions, where now we can see that we can identify genes that are upregulated or switched on in both the healthy state, but also genes that are upregulated or switched on in the type 2 diabetes state. And again, you can see how genes that are potentially switched on or differentially expressed in diabetes would be potential candidates for drug discovery. I'd now might like to move on to how we can think about discovering novel small peptides and using those in drug discovery. So in order to discuss this, we're going to return back to this process of translation, which was generating proteins from our intermediate RNA. The code on this RNA, which encodes for this protein, can be termed an open reading frame, or an ORF. Now, traditional annotation of these ORFs, and therefore their proteins, has typically excluded small open reading frames that are fewer than 100 amino acids. Remembering amino acids are these building blocks of the protein. And this was typically due to the high number of predicted small open reading frames and the now incorrect belief that they were non-functional. But we know that the 21,000 protein coding genes that are traditionally defined actually only represent 1% of the human genome. So we know there is a huge amount of potential there that is still underexplored. We now know that there are potentially millions of small open reading frames, or SMORFs, that are found throughout the human genome, and many are thought to encode novel SMORF-encoded peptides. In humans, we now know that these small open reading frame encoded peptides are both functional and biologically relevant, and they've been shown to play a role in a range of processes, including transcriptional regulation, so that was the process of generating RNA from DNA, muscle contraction, and cell growth, just to name a few. But how do we study and discover these small open reading frames and their proteins? In order to answer this question, we need to turn back to the process of translation in a little bit more detail. The ribosome is the translational machinery found in all our cells. When an mRNA is ready for its instructions to make a protein to be utilized, a ribosome will bind this RNA. This ribosome then enables the production of a polypeptide or protein to be generated from the mRNA. So we can see that the mRNA that is bound by ribosomes represents the proteins and the peptides that are currently being translated or generated in a cell. And we can utilize this information. And this is through a process known as ribosome profiling or ribosome sequencing. In this process, we identify the ribosome protected mRNA fragments So this is the mRNA that is being bound by ribosomes because protein is being generated from it. And these are termed ribosomal footprints. These ribosome protected fragments, or ribosomal footprints, identify the position of mRNA that is being engaged in translation. And this then enables us to decode this into a profile of proteins that are being generated in a cell. And this is really a transformative technique to look at global analysis of this translatome, or the translated proteins. So how do we use this to be able to discover new peptides and look at how they may be relevant to disease? To do this, we can start with human tissue, and we can then isolate these mRNA protected ribosomal fragments, and we can carry out ribosomal profiling. As we've just seen, this allows us to come up with a profile of novel small peptides. But how do we link this to disease? And to answer that, we're going back to our RNA. You can see if this is our RNA, and this is our small open reading frame that encodes for our small open reading frame encoded peptide. But if we go one step up, we can actually go back to the DNA that is encoding this small open reading frame, and we can look for the presence of different genetic mutations in this DNA. For example, we can look for the presence of genetic mutations that may be associated with type 2 diabetes. As we saw with the factor VIII example, a genetic mutation in the code for a small open reading frame will potentially have a functional effect on this small peptide. And by identifying morphs that are overlapping with genetic mutations that are associated with disease, you may be able to identify these peptides that are playing a role in disease biology. And therefore, we can see how these may be candidates for drug targets. Lastly, I'd just like to share some of the thought processes which we need to go through when we're thinking about prioritizing these targets in drug discovery. As you've seen, we can use a range of methods to try to identify genes or proteins that may be associated with the disease. However, many of these methods generate several numbers of lists of genes and proteins associated with disease, and we need a way in which we can approach and think quite carefully about the ones we decide to take forward in drug discovery. And we can split this up into four main questions, which include what, where, how, and who. If we start with what, we need to think about what gene or protein do we want to target. In order to answer this, we think about building multiple layers of evidence to support this disease association. And this is often a combination of both some of the computational methods that I've shown you and others, but also working very closely with our colleagues in the wet lab to generate experimental support for these drug targets. We can then move on to asking ourselves, where in the body is this target found? Which tissues are expressing this gene or protein? Can we target that tissue? Can we predict any side effects due to expression in other tissues? This is extremely relevant when we're thinking about how to move it forward. And it is also extremely relevant when we move on to our third question of how, which is how can a drug be designed? And to answer this, we need to think about not only the different drug modalities. Are we going to use a small molecule to target it? Are we going to use gene therapy? But also the drug formulations of, will this be an oral tablet? Will this be an injectable? And finally, we can move on to the who, which is who would benefit from the drug. Using the large-scale clinical data that we have access to, can we try to predict patient response to our drug? And can this be used to try to develop a precision medicine approach to really get the right drugs to the right people? And with that, I'd like to finish, and I'd like to thank you for your time and I'm more than happy to answer some questions.
Thank you very much, Zahra. It was a really interesting talk, and I'm glad we managed to get those sound issues sorted out as well so we could actually hear it. Okay, so yes, if anyone watching has got any questions, please do pop them in the comments box and they will come through. Committee members, if you have any questions, please wave at me from your green room boxes and I can bring you in. I was wondering, Zahra, if you could pick up a little bit on something from your final slide where you were talking about being able to predict patient response based on existing data sets. That obviously is something, if we could do that accurately, that would potentially reduce the amount of time needed for trials because things could be more targeted. Could you maybe expand upon where we are with that in reality rather than the goal?
Yeah, no, it's a great question. I think there's applications both in improving our clinical trial design, but also in a real world setting where we're thinking about not putting people on drugs where there's no need and it won't necessarily be something they'll respond to, but also trying to identify people who would really benefit from something. And I'd say we're actually fairly close to being there. There is the ability to work with, for example, electronic health record data, where we can look at drugs that people have been prescribed. We can look at some lab results they have in terms of what is the reaction in their level of cholesterol, what is their blood sugar like, and we can see how the drug is having an effect on these parameters. And we can also look to see how they may develop other complications of the disease. So, for example, in type 2 diabetes, we can say, is this person on this drug actually still going on to develop cardiovascular disease as a side effect, or is it actually helping to prevent that? And we have that data, and we're now starting to get some methods, including some machine learning methods that we're using at the moment to try to really study that and see how we can actually characterize these groups of individuals and identify something that we can actually then transfer into the clinic and say, actually, if you have these characteristics, you may respond well to this, you may not respond well to that. And I think it's that transferring it to the clinic is, I think, where we're at. I think we need something that's very robust and been validated before we can take it there. But I think the data and the methods are actually getting there now.
That's really cool. Interesting you're saying about how the use of electronic patient records. Does this potentially mean that a rate limiting step is the different laws surrounding data ownership and ownership of one's medical records in different countries? I know when with DNA sequencing, when that first became more accessible, there was a big thing over in the US, especially over who owns a DNA sequence from something that's in the public domain. So is that an issue, or has it not been a problem?
Another really good point, and I think something that's really helping that area advance is an initiative called Odyssey or OMOP, which is a group which has really got the mission of firstly mapping all these different electronic healthcare data sets and medical data sets we have to a common structure and data model, which enables us to do more repeatable analyses. And also something which them and others are thinking a lot about is this concept of a federated database, which is where actually the data itself never leaves where it has been generated. So we're able to maintain this privacy, we're able to maintain anonymity, and we're able to not think about exporting data. But there is a layer in which you can apply methods to this federated database, and you can pool the data together in this federated database. And that enables you to really increase your numbers as well, which is great for discovery. So I think there's a lot of methods we can try to use to still maintain privacy and anonymity while also enabling some of these data to become accessible for research.
Cool. That's really interesting. Thank you very much for that. Does anyone in the committee? Oh, I have a question coming through. Second. So Leonardo asks, he says hello, and asks, can you also look at transcriptomics using the same cell types at different times of the cell cycle?
It's a great question. And especially in the world of single cell transcriptomics, the cell cycle is something that can play havoc with that data. Because of course, if you imagine I take 100 cells they're all going to be at a slightly different place. And same if we took 100 people and they're all in a slightly different place in life, all these cells are at a slightly different place in their cell cycle. And if you take a snapshot of that transcriptomics and it's just frozen in time, then you're going to see evidence in that in all the genes that they're switching off and on. And oftentimes that's not what people want to look at, and they actually try to correct for those differences. So you can actually then look at what are the different cell types, what are the differences across disease. But you can absolutely specifically try to take cells that are at different stages of the cell cycle and then look at the genes they're switching off and on. And it's definitely something that can be used there.
Yeah, I know it's a bit easier if you're working with cell lines, I know, because you can arrest and have the cells all at specific points. But I guess it's a bit harder and more nuanced with tissue samples and patient samples in that way.
Yes, yeah, a little bit more delicate, but
Cool, thank you very much. Does anyone in the committee have a question? Not yet. Okay. I've just been handed a question. Those of you who tuned in January will recognize my companion this evening. So Demetrius asks a long question. Sequencing data offers only a static window in disease, and diseases are incredibly dynamic rather than static. How would you overcome the difficulty in sample acquisition and patient follow-up, especially in invasive acquisitions such as solid biopsies?
Yeah, it's, again, a very good point. Of course, diseases are extremely progressive and changing with time, and that's something that more and more we're trying to take into account. And working with longitudinal data where you have samples from follow-ups is something that is happening, I think, more and more. And thinking about how you model this time series data is something which is also developing. In terms of avoiding invasive biopsies, again, that's something there is a lot of interest in, especially when you look towards a clinical trial setting where, of course, we don't want to put unnecessary burden on patients undergoing those trials. And that's where you really start to try to identify what we call biomarkers in either the blood or urine or something else that's more easily accessible. And that can be something that, for example, if your drug is acting in the liver and you want to look for your drug doing a specific thing in the liver, you don't want to have to keep on taking liver biopsies. So instead, you try to identify a circulating biomarker in the blood that is very well correlated with what your target's doing in the liver. And that way, it's a much lower patient burden, and you just need to go in and take a small blood sample, and you get this correlation, which tells you what's probably going on in the liver. And that is something which, even in Novo Nordisk, I know we have a high focus on at a very early stage when we assess drug targets to also be looking for these different types of biomarkers.
Thank you. We have a couple of questions from the committee, so I can't bring them up on the screen, but Susan asks, are all genes expressed in human DNA, or is it a specific percentage expressed?
Yeah, it is definitely a certain percentage is expressed, and that will be different across different cell types. So in, for example, our beta cell and our alpha cell, they will go to their DNA and they'll say, what do I actually need from here to do my job? I'm only expressing you. And it'll be that 5%. That's a made up number, but it'll be that 5%. Then you'll have your alpha cell, which will go and look at its DNA and say, actually, to do my very different job, I need this set. And that will be a different 7%. So it's a dynamic process in that it's actually very well regulated and finely tuned to what's going on around it to respond to different stimuli. But to go in and turn, and of course, it's not as simple as switching a gene off and a gene on. There's some granulation there where you can go and turn something up, turn it down. And so it's a very finely tuned process of responding to what's going on and what it needs to do.
Thank you. Peter asks, relating back to the data integrity conversation we were having earlier, I think he asked, does Novo Nordisk work with anonymized information from the UK Biobank?
Yes, and I can say yes to that. And I personally work with UK Biobank quite a lot. For those who are not familiar with it, the UK Biobank is a resource of about 500,000 people from the UK who have very kindly volunteered to take part in this initiative, where they answer numerous questions about their health, their medical history, they donate samples, they have gone through this process of genome-wide genotyping that I described, And it's actually a world leader in setting the example for how this type of resource for research can be done. It's very obviously restricted access to researchers doing bona fide research, but they make it very accessible to researchers, especially in academia where funds may be sometimes lower. They actually have a very fair costing model to enable people to do amazing research, and that's actually something that it's enabled it to be used worldwide to help a lot of different fields.
Thank you very much. No? Okay. Okay, I think that was all of the questions that I had that came through. So thank you very much again, Zahra. It was a really interesting talk and a really nice discussion afterwards. So thank you for coming along and speaking to us this evening. Just a real quick final thing to wrap up, because I did say that I'd bring them up if anyone put any extra science snippets on. Bob did pop a comment in to say that lab experiments published last week indicate that 3 billion years ago, the planet's oceans housed nearly twice as much water as now, which affected plate tectonics and how life starts on land. So there you go, and we'll go look for that story elsewhere. Cool, thank you very much. Thank you all for coming along this evening, and we'll see you next month.
Thank you.
Bye.
Bye.